Now, I'm not going to show the phenotypes of the flies because I just showed them to you, but in the next in this module we're going to show the way in which meiosis gives you the frequencies of progeny in the case of linked genes. So in this particular case, what we did is we took a cross and we had an individual. Now I'm not showing colors anymore. Little PR, okay, little VG, okay. Was crossed with an individual that was wild type for both and homozygous, okay. All right, and we created this individual then, okay. The only outcome of this. Okay, now this individual appears wild type because these are recessive, and then we cross that with this particular individual here as a test cross. And we asked, what are the outcomes of this? So what we're going to do is we're going to talk about what are the different types of gametes that can come out of this meiosis, and why do they come into the particular frequencies that we expect. If we have 10 centimorgans here, okay, that'd be 10% recombinants in the recombinant class, okay. So about 1,000 progeny, you would get gametes, you know, not quite half the time coming from this and not quite half the time coming from this, because you would get some of the recombinants in there. So what does meiosis look like in this individual to produce gametes that we transmit to the and cause fertilization with the gametes of this individual? Okay, so we're going to do that meiosis right now. Okay, so this individual has this particular chromosome assortment. Okay, we can talk about chromosomes from dad and chromosomes from mom. We're not going to quite do that right now. Putting some long sister chromatids there. Okay, so this individual is PR, big PR, and big vestigial wild type, and then the mutant alleles, little PR here, and then little VG here. Okay. Now this, of course, is after DNA replication. Okay. Now some percentage of the time, okay, these things will recombine in meiosis, and recombination happens, or well, let me just be more clear about this. Crossing over happens in the two-strand stage, and we can think about how we know that in a moment. Okay. Now, when you have crossing over, basically what's going on is there's an exchange of flanking markers. So you're going to get a crossover here. Okay. So this particular crossover would produce what kind of meiosis? Okay. Well, actually, if you think about it, only 2 sets of chromatids have crossed over with one another. These other 2 don't. Okay. So there's a crossover in this meiosis between them. Okay. But only half of the chromatids that actually participate in this crossover reaction. Okay. So what's going to happen is these are going to cross over, and you're going to basically end up with chromatids that look like this. Okay. After the crossover is resolved, so these ones didn't participate in crossing over. That's this big PR over here, but now little vestigial right here. Okay. And then little PR right here, but big vestigial over there. Okay, and then now these are still the recessive alleles, mutant alleles. Okay. So what does this end up doing? Well, there end up now, and before, without crossing over or anything like that, you ended up with basically a single gene, of course. You ended up with 4 different haploid products, 2 of which are identical. So in the case of you have big A possibilities or little a possibilities, in this particular case you end up with 4 gametes. Okay. Now, what are those possible gametes? Well, they segregate away from each other in M1, and then they segregate and split at the centromeres in M2, and you end up with these particular gametes. Okay. Haploid, of course, big PR, big VG. Okay. That's corresponding to this one here. Okay, and then you also get big PR, little VG, okay? And then you get the corresponding reciprocal of this one here, which is little PR, big VG. Okay, and then finally you end up with little PR, little VG. Okay. Now, in this particular case, these are basically what you would call the parentals, and these would be what you would call the recombinants. Now, what's important here is you had one meiosis, okay? One crossover here, and half the products of recombinant. So this is a recombinant, and this one here is a recombinant. Okay. Now, the frequency at which this occurs, okay? Suppose, for example, in the case of purple and vestigial, we said that they're 10 centimorgans apart. What that means on average is that about 20% of the time there's a crossover somewhere between there, okay? 20% of the time there's a crossover, but of those myocytes, of those meiotic products that have crossed over, only half of them produce what looks like a recombinant progeny, okay? So in this particular case, you can think about it that way. Now, and the next thing that we're going to do is we're going to actually sort of use meiosis in considering another gene that's not linked to either of these ones and finish up the visualization of meiosis with recombination and show you how alleles segregate in that fashion.